Okay, we need to kind of think about all that we've talked about with regard to cells, the fundamental unit of life so far. We've talked about quite a lot. We have talked about um, cell membranes and all the important things that they can do, the way things pass across them. So we've talked about um, channels that allow diffusion to move across. We've talked about pumps and the importance of that. We've talked about proton pumps and the mitochondrion. We've talked about the sodium potassium pump in the neuron. We've talked about receptors in cell membranes. We've talked about that in terms of the immune system and how a B or a T cell might dock with a ID marker on a bacteria or something like that. We've talked about receptors and how they are used in the endocrine system to cause a response. We've talked about receptors in the nervous system. So we've talked about a whole lot of stuff just with regard to cell membranes. We've talked in detail about the mitochondrion. Whoops. And how that works to make energy or transform energy for the cell to use. One thing we haven't talked about so far, though, and that we really need to, is how cells reproduce. How do cells reproduce? Make more cells. This is going to be, obviously this is just an important thing in and of itself, but it's going to be very important for us as we begin to approach our genetics unit. So we're going to talk about cellular reproduction, okay? And what we talk about when we talk about cellular reproduction, generally speaking, first is we talk about something called mitosis, and we also talk about something called the cell cycle. Okay, so that's where we're headed with what we're going to talk about in this podcast. So we've talked a whole lot about cells, and now we're going to talk about how cells make more cells, okay? And this will highlight some other features of cells, parts of cells that we haven't yet talked about. So, that said, let's talk about the cell cycle. All right. To set this up for us, imagine your skin, okay? You've got skin. This is the very top of your skin that you would see and touch, and this is, generally speaking, made out of dead cells, okay? And down at the base of your epidermis, by the way, that's what this whole set of skin layers is called. All of this is epidermis. It's made up of essentially one, two, three, four, 5 layers that are distinct. This bottom layer is the part where cells are reproducing. Okay, the stratum basal is where the cells are reproducing. Okay, so we got lots of cellular reproduction, making baby cells here. Okay, by the time these skin cells migrate their way up to the top, they essentially are dead, and then they begin to slough off the top, which is why we keep having to make more down here at the bottom, because we keep losing them. They slough off at the top. You lose skin cells every day. It's just a part of your life. So to replace what you lose, we have to keep growing them from the bottom, and then they migrate up towards the surface. So down here in this bottom layer of your epidermis, Mitosis is happening all the time. We are creating new skin cells there. And we don't want the skin cells that are made new to be any different from the skin cells that were there in the first place. We want them to be just the same. We want them to be your skin. So we want to make exact copies. We want to make exact copies of skin cells. Okay? We want duplicate cells. That's really important for everything we're about to talk about. And this particular podcast has to do with how we make duplicate cells that are exactly like a parent cell. We are talking asexual reproduction here, okay? So, in order to live, cells have a life cycle they go through. And interestingly, it goes from birth to birth to birth to birth. No death in there. Okay? How can that work? Is there death? Eventually, cells do die. Most human cells, at least, are limited in the number of times they can go through reproduction. They can give rise to new cells. For example, if you put infant cells in a petri dish, they may reproduce 100 or more times, but then eventually they will stop reproducing and eventually they'll die. If you put skin cells from a 60-year-old, they won't reproduce near as many times because they've already used up many of their allotted reproduction events, and they'll only reproduce maybe 20 times before they die. So there's a finite amount of birth events that a cell can have where it can copy itself and make new cells. But until they hit that ultimate limit of 100 times that they're allowed to divide, they go through this birth, they live a while, then they give birth to more cells. They go through this cycle over and over. So, if it goes from birth to birth to birth to birth, what's in between each cell birth event? Well, of course, life. The cell lives its life. The cell carries out its life functions, okay? It does whatever job it's supposed to do. If it is a skin cell, for example, it's a skin cell. It does its job being a skin cell. And when it's time, it will produce more skin cells. And then they will do their jobs of being skin cells, and so on and so forth. Now, this life cycle of a cell, from the point where we have a cell be born, I'll represent that at the X, and then the cell lives its life, but it comes around and decides, oh, I've got to give rise to more cells, I've got to give birth to more cells. Then we have another birth event, and we have another life cycle until we have another birth event. This is called the cell cycle, okay? Now, by convention, we normally talk about a stage in the cell's life called interphase, and you may remember that from 9th grade or before, as the stage that marks the beginning of the cell cycle. And I should have put a close quote, okay? It's a cycle, so you can start anywhere in that cycle, but we'll call interphase the beginning because that usually makes it easier, okay? Now, interphase is the stage in a cell's life where it is basically doing one of two things. It is living out its life, meaning if it's a skin cell, it's being a skin cell, or it is preparing to make baby skin cells. It is preparing for cell division, the birth of two new cells. Those are the two events that happen in interphase. The cell does its job, or it's getting ready to make more cells. Okay? Now, interphase is the vast majority of the cell's life. I mean, let's think of it. It's where the cell is doing whatever it's supposed to do. It's being a skin cell. So if the cell cycle, the cell's life, is a great big pie, there is a very thin slice of pie represented right here, which is actually the cell dividing and making babies. The vast majority of the cell's time is spent being a skin cell, or whatever kind of cell it's supposed to be, okay? Doing its everyday job. Now, if it gets a signal from its surroundings that it's time to divide, it's time to make new cells, then it will begin making preparations to do that. And then it will actually make the new cells during this very small window of time right there called mitosis. Okay? So, the cell cycle then is the life cycle of the cell, and interphase is a huge chunk of it. And interphase has some significant things, even though we often think of it as not doing anything because it's the cell's not actively dividing. It is still doing stuff that is necessary. So once a cell is born, so let's say that's our cell birth event right there, it grows a little bit. So let's say this is where the cell is born, right there, where this red and blue arrow meet. During this time, the cell is going to grow in size a little bit, and it's going to go through some maturation, okay? And this is called the G1 phase of the cell cycle, or gap 1, and I once knew why they call it that, and I can't remember, okay? And what happens during this time is we are preparing for division, okay? Now, once we divide, then we would have two cells that would be doing this, and then they would divide, and so on. There are some cells, however, that leave the cell cycle, that don't keep repeating it, don't keep giving rise to cells that will divide and divide again. Some cells grow and then they leave the cell cycle, so to speak, and don't divide anymore. Cells that do that, notoriously, nervous system cells are notoriously stagnant once they have formed. They don't now we know there is some suggestion that nerve cells really can divide if they are damaged, but it is not something that is well understood, which is why spinal cord injuries can be so devastating. If you damage nerve cells, they don't normally regrow in ways, but we also know that they can. We used to think they could not at all, and now we know they can, and the idea is to figure out how to harness that and trigger them to grow when someone has had an injury. Muscle cells are notoriously very hard to encourage to grow once they have stopped forming during development. Liver cells, generally speaking, don't divide again once they formed, but they can be coaxed into it if the liver is damaged. So there are some cells that kind of go out and go resting until there's some reason for them to come back and start dividing. So there's all kinds of caveats with this, but generally speaking, this is the cell cycle that we're going to be talking about over here, and just understand that some cells can leave that cycle. So interphase is divided into three parts. As I said, G1. G1 is where the cell is basically doing its job. It's doing whatever it's supposed to do to be a cell. It is growing a little bit bigger, because remember, when a cell starts out, it is small, so it will increase in size and get a little larger over time. It does that during the G1 phase. Okay, then for whatever reason, it detects a signal that tells it to divide. At this point, the DNA, the genetic material in the cell, is going to copy itself exactly. Now we need to think about why this is. Remember, what we are talking about right now is asexual reproduction. We are talking about we have skin cells and we want to make more skin cells that are exactly like those skin cells that we started with. We want exact copies. This means that if this is my DNA right here, Okay? I can't just chop that DNA in half and say, okay, here's for one cell and here's another cell, because then these two cells only have half of what I had to start with. They have half the genetic information. That's no good. They have to have all of the genetic directions for how to be a skin cell. So what I have to do then, if this is my genetic material and I'm the parent skin cell, okay, I have to be able to make another duplicate copy exactly like that copy. And I have to be able to give a full copy to each of my daughter cells. Okay, so that's why we have to do this step right here. The DNA has to exactly duplicate itself. Okay, so we exactly copy the chromosomes. Then we make our final preparations to divide during the G2 phase. Once we've duplicated our DNA, we make grow a little more, make our final preparations for cell division, and then we will begin dividing. Okay, so interphase is not stagnant. You need to know that. Interphase has got G1, S, and G2 phases, and this S phase is of particular importance because the DNA, the genetic material, the chromosomes, whatever you want to call it, it's all the same, is going to be copying itself. We call that replication, and I cannot overemphasize to you the importance of that word. We're going to talk about it quite a bit. And also remember, if the cell is not destined to divide, it can leave the cell cycle and kind of go into GO, or a resting state. Most nerve cells, muscle cells, and so on would be in that state. Okay? Now, we'll talk about more of this later. But there are certain checkpoints. Here is a checkpoint mentioned in the G1 stage, okay, where the cell will actually check itself. It's a control mechanism so that we can be sure that everything is ready that needs to be ready for the next stage of the cycle, which is the synthesis stage. We will have another checkpoint here, right there, and the second gap, the G2 phase, to make sure everything's okay to enter mitosis, to actually divide. And these checkpoints are really to make sure that everything is proper within the cell, so that the cell, we know the cell is not diseased, it's not cancerous, it's not mutated to the point where it's going to cause us harm. So this is really, really important, because if things do not check out either here or here, the cell will be arrested and stopped at that point in its development, and it won't be allowed to copy itself anymore. So that's really, really important. It's a way that the body has of checking to make sure we don't have cancerous cells that are going to get out of control. Obviously, sometimes cancer cells are able to evade these controls, and that's when we have a problem. All right. All right. Now, before we go into mitosis, it's really important to understand how the chromosomes are built and how they change after they've been replicated, what they look like. So during G1, during interphase in G1, it's important that you understand first that the chromosomes are long and stringy, okay? And I might have 6 chromosomes in this cell, but they're all long and stringy, they're all mixed together, and it's really hard to tell the difference, okay? Now, let's see. Erase all ink on slide. That's what I wanted to do. Now, I'm going to focus on just one of these long, skinny chromosomes right now. Okay. It looks like this during G1. Long, skinny, and when the chromosomes are like this, we call them chromatin. Okay, they're thin and stringy, and if you looked at a cell under a microscope when it was in G1, here's the cell, the nucleus would basically just be this blob in the middle. You couldn't really see that there were chromosomes in there because they're just all like a plate of spaghetti. You can't really tell one noodle from another in a plate of spaghetti. They're all tangled up. Now, during G1, this is how our chromosome would look. Okay? Now, during S, when the cell is ready and is definitely replicating, this is where replication of the DNA occurs. We have this one long strand of DNA. That's what the chromosomes are, remember, is a strand of DNA. We have one long strand of DNA. If we super magnified it, okay, we'd see that traditional double helix. Okay, so this is from G1. Well, during S, this one long strand is going to replicate itself exactly. It's going to replicate itself exactly, and it is going to be right next to the original, and they will actually be joined at a spot somewhere around the middle that is called a centromere. Okay? Now, each of these strands is identical to the other. Identical, exactly the same. Okay? We've just made an exact copy so that when we reproduce, when we make baby cells, each of them is going to get a copy of it. So, this strand is called a sister chromatid, as is this strand. They are made up. So this is, getting our terminology straight, one chromosome still. It just happens to be double-stranded, and it is made up of two sister chromatids. Okay? So that happens during the S phase. We have duplicated that one long skinny DNA molecule, and now we have two long skinny DNA molecules attached at a centromere. Okay? Now, at G2, then, basically, we've still got our replicated chromosomes, if you will, that look like that, but now the cell is just making all of its final preparations to begin dividing. Okay? All right. Now, after all of that, we are ready to give birth to new cells, and we are ready to enter into actual mitosis. And I'm going to stop the show here, and then we will start it up again with part B, which will be actually dividing the nuclear material and then the cell itself. Okay? Thanks for watching, and we will see you next time.